Dear students, next topic is nucleotide metabolism. Before going to the topic, please subscribe my channel, press the bell icon for notifications, like, share among your friends and comment. Dear students, nucleotides are the building blocks of nucleic acids. You know, they are of two types, deoxyribonucleic acid or DNA and ribonucleic acid or RNA. The most basic structure of the nucleotide can be broken down into three subunits. First one is a 5 carbon sugar, ribose or deoxyribose, a phosphate group and a nitrogenous base, also known as nucleobase. So, the 5 carbon sugar is either deoxyribose or ribose, and depending on which is used, the final product is either deoxyribonucleic acid or ribonucleic acid. The nucleobases can be either purines or pyrimidines. There are three types of pyrimidine bases and they are all made up of single heterocyclic ring like cytosine or C, thymine or T. Thymine is DNA specific and uracil or U, which is RNA specific. Purine bases are of two types, adenine or A and guanine or G. They are made up of two rings. Now, if we link up just the sugar and the nucleobase, we have got ourselves a nucleoside. To make a nucleotide, all we have got to do is add a phosphate group to the fifth carbon of the sugar on a nucleoside. So, nucleosides have slightly different names. In RNA, ribose plus adenine makes adenosine, guanine makes guanosine, cytosine makes cytidine and uracil makes uridine. So, adding a phosphate, the full name of RNA nucleotides would actually be adenosine monophosphate or AMP, guanosine monophosphate or GMP, cytidine monophosphate or CMP and uridine monophosphate or UMP. For DNA, we are using deoxyribose instead of ribose. So, the nucleosides would be deoxyadenosine, deoxyguanosine, deoxycytidine and deoxythymidine, and similarly with the addition of phosphate group, the nucleotide would be called, for example, deoxyguanosine monophosphate or DGMP like. There are two ways our cells can make nucleotides. One is to make from scratch, also known as de novo synthesis, and the other is the salvage pathway that recycles nucleotides that are already semi-degraded. De novo synthesis starts with the ribose 5-phosphate for both purine and pyrimidine bases. Let us begin with the purine synthesis, that is biosynthesis of purines. Ribose 5-phosphate come from another intracellular metabolic pathway called the pentose phosphate pathway, and an enzyme called ribose phosphate pyrophosphokinase uses an adenosine triphosphate or ATP molecule and removes two phosphate groups from it, attaching them to ribose 5-phosphate, creating phosphoribosine pyrophosphate or PRPP. Synthesis of purine. Synthesis of purines take place in 10 steps. See, this is the diagrammatic representation of purine synthesis and each steps are explained from the following slides. First is the formation of PRPP, addition of N9, addition of 4th carbon, 5th and N7, addition of C8, addition of N3, cyclization, that is closure of the ring, addition of C6, addition of N1, removal of fumarate, addition of C2, cyclization, then the formation of these adenosine monophosphate and guanosine monophosphate. See here in the first step, ribose 5-phosphate produced in the hexose monophosphate shunt, that is HMP shunt, of carbohydrate metabolism is the starting material for purine nucleotide synthesis. It reacts with ATP to form phosphoribosyl pyrophosphate, that is PRPP. PRPP synthetase is inhibited by PRPP. In the second step, glutamine transfers its amide nitrogen to PRPP to replace pyrophosphate and produce 5-phosphoribosylamine. The enzyme PRPP glutamyl amidotransferase is controlled by feedback inhibition of nucleotides, that is IMP, AMP and GMP. In the third step, phosphoribosylamine react with glycine in the presence of ATP to form glycinamide ribosyl 5-phosphate or glycinamide ribotide GAR. In the fourth step, NS N10 formyl tetrahydrofolate donate the formyl group and the product formed is formyl glycinamide ribosyl 5-phosphate. In the fifth step, glutamine transfers the second amino group to produce formyl glycinamide ribosyl 5-phosphate. Then the imidazole ring of the purine is closed in an ATP dependent reaction to yield 5-amino imidazole ribosyl 5-phosphate. The seventh step, incorporation of carbon dioxide, that is carboxylation, occurs to yield amino imidazole carboxylate ribosyl 5-phosphate. This reaction does not require the vitamin biotin and for ATP, which is the case with most of the carboxylation reaction. In the eighth step, aspartate condenses with amino imidazole carboxylate ribosyl 5-phosphate to form amino imidazole 4-succinyl carboxamide ribosyl 5-phosphate. In the ninth step, adenosaccinase cleaves off fumarate and only the amino group of aspartate is retained to yield amino imidazole 4-carboxamide ribosyl 5-phosphate. In the 10th step, N10 formyl tetrahydrofolate donates a 1-carbon moiety to produce imidazole 4-carboxamide ribosyl 5-phosphate. With this reaction, all the carbon and nitrogen atom of purine ring are contributed by the respective sources. And in the 11th step, the final step is catalyzed by cyclohydrolase, lead to ring closure with an elimination of water molecule from formimidazole ribosyl 5-phosphate by inosine monophosphate IMP. Cyclohydrolase forms IMP. By inosine monophosphate, cyclohydrolase forms inosine monophosphate. See here, synthesis of AMP and GMP from IMP are shown. Next is the salvage pathway for purines. What is salvage pathway? Most cells have an active turnover of many of their nucleic acids, particularly some types of RNA, which result in the release of adenine, guanine and hypoxanthine. These free purines are reconverted to their corresponding nucleotide through salvage pathways. See here, adenine phosphoribosyl transferase convert to AMP, adenosine monophosphate, then hypoxanthine to inosine monophosphate, then to guanine in presence of PRPP, GMP, guanosine monophosphate. How formation of AMP occurs? Adenine plus ribose 1-phosphate in presence of nucleoside phosphorylase converted to adenosine plus inorganic phosphate. Then here one ATP undergo hydrolysis and form AMP, adenosine monophosphate and adenosine diphosphate in presence of the enzyme adenosine kinase. Regulation of purine nucleotide biosynthesis. Purine nucleotide synthesis is well coordinated to meet the cellular demands. The intracellular concentration of PRPP regulate purine synthesis to a large extent. This in turn is dependent on the availability of ribose 5-phosphate and the enzyme PRPP synthetase. PRPP glutamyl amidotransferase is controlled by a feedback mechanism by purine nucleotides. If AMP and GMP are available in adequate amount to meet the cellular requirements, their synthesis is turned off at the amidotransferase reaction. Another important stage of regulation is in the conversion of IMP to AMP and GMP. AMP inhibits adenylosuccinate synthetase while GMP inhibits inosine monophosphate dehydrogenase. Thus, AMP and GMP control their respective synthesis from IMP by a feedback mechanism. This is the interconversion and regulation of IMP to adenosine nucleotides and guanosine nucleotides. Formation of nucleotide from nucleoside di and triphosphates by ATP. See here, nucleoside monophosphate will undergo reaction in presence of kinase by utilizing ATP, convert into nucleoside diphosphate. Again, kinase enzyme will act on it in presence of ATP and form nucleoside triphosphate. Conversion of nucleoside monophosphate to nucleoside diphosphate and triphosphate is shown in this equation. This is the conversion of ribonucleotide to deoxyribonucleotides. The synthesis of purine and pyrimidine deoxyribonucleotides occur from ribonucleotides by a reduction at the C2 of ribose moiety. This reaction is catabolized by enzyme ribonucleotide reductase. The enzyme ribonucleotide reductase itself provides hydrogen atoms needed for reduction from sulfhydryl groups. The reducing equivalents in turn are supplied by thioredoxin, a monomeric protein with two cysteine residues. NADPH dependent thioredoxin reductase converts oxidized thioredoxin to reduced form, that is shown in this figure. How degradation of purine nucleotide occurs? The end product of purine metabolism is uric acid. The nucleotide monophosphate, that is adenosine monophosphate, AMP, inosine monophosphate, IMP, and guanosine monophosphate are converted to their respective nucleoside forms, that is adenosine, inosine, and guanosine by the action of nucleosidase. In the second step, the amino group either from AMP or adenosine can be removed to produce IMP or inosine respectively. In the third step, inosine and guanosine are respectively converted to hypoxanthine and guanine, that is purine bases, by purine nucleoside phosphorylase. In the fourth step, adenosine is not degraded by this enzyme, hence it has to be converted to inosine. Guanine undergo deamination by guanase to form xanthine, and finally xanthine oxidase converts xanthine to uric acid. Uric acid 268 TTI oxopurine is the final excretory product purine metabolism in human. Uric acid can serve as an important antioxidant by getting itself converted non-enzymatically to allantoin. It is believed that the antioxidant role of ascorbic acid in primates is replaced by uric acid, since these animals have lost the ability to synthesize ascorbic acid. Most animals other than primates, however, oxidize uric acid by the enzyme uricase to allantoin, where the purine ring is cleaved. Allantoin is then converted to allantoic acid and excreted in some fishes. Further degradation of allantoic acid may occur to produce urea in amphibians, most fishes and some mollusks, and later to ammonia in marine invertebrates. Dear students, next step is to make pyrimidines or biosynthesis of pyrimidines. Compared to purine, the biosynthesis of pyrimidine is much simpler process. Aspartate, glutamine and carbon dioxide contribute to atoms in the formation of pyrimidine ring. Pyrimidine ring is first synthesized and then attached to ribose 5-phosphate. This is in contrast to purine nucleotide synthesis, wherein purine ring is built upon a pre-existing ribose 5-phosphate. Here also there are 11 steps involved. In the first step, formation of carbamyl phosphate. Carbamyl phosphate is formed from ATP, glutamine and carbon dioxide. The reaction is catalyzed by carbamyl phosphate CPS2. The second step is the condensation. Carbamyl phosphate condenses with aspartate to form carbamyl aspartate, catalyzed by aspartate transcarbamylase. Third reaction is ring closure. This occurs via loss of water. This reaction is catalyzed by dihydroorotase, forming dihydroorotic acid. Fourth step is dehydrogenation, removal of hydrogen atom from 5th and 6th carbon by dihydroorotate dehydrogenase, that from mitochondria. The fifth step is transfer of ribose phosphate. This is transferred from PRPP forming OMP, that is orotidylate, catalyzed by orotate phosphoribosal transferase. The sixth step is decarboxylation. OMP is decarboxylated forming UMP. UMP is the first true pyrimidine ribonucleotide. The seventh step is phosphorylation of UMP forms UDP and UTP with the help of ATP. And the eighth step is formation of CTP, cytidine phosphate. UTP is aminated by glutamine and ATP, catalyzed by CTP synthetase. The ninth step, reduction of ribonucleoside diphosphate to their corresponding dinucleodiphosphatase. That is to their corresponding DNDPS. 10th step, formation of TMP from UDP. DUMP is substrate for TMP synthesis. DUDP is dephosphorylated to DUMP. The 11th step is methylation of DUMP. This occurs at the 5th carbon by 5th nitrogen, 10th nitrogen methylene THF forming TMP. This reaction is catalyzed by thymidylate synthetase. All these reactions are shown schematically. Here is also salvage pathway of pyrimidine synthesis, that is interconversion of pyrimidine base plus PRPP to pyrimidine phosphoribosyl transferase will form pyrimidine nucleotide plus inorganic pyrophosphate. Here is a regulation of pyrimidine synthesis. CPS2, aspartate transcarboamylase and dihydroorotase are present as multi-enzyme complex. Orotate, phosphoribosyl transferase and OMP decarboxylase are present as single functional enzyme. Due to clustering of these enzymes, the synthesis is well coordinated. The hydroorotate, dihydroorotate dehydrogenase is mitochondrial enzyme. CS2 and aspartate transcarboamylase and OPRT transferase and OMP decarboxylase are sensitive to allosteric regulation. CPS2 is main regulatory enzyme in mammalian cell. CPS2 inhibited by UTP, activated by PRPP. Aspartate transcarboamylase, main regulatory enzyme in prokaryotes, inhibited by CTP, activated by ATP. Requirement of ATP or CTP synthesis and stimulatory effect of GTP on CTP synthetase ensures a balanced synthesis of purines and pyrimidines. Here is the degradation of pyrimidine nucleotides. The pyrimidine nucleotides undergo similar reactions, dephosphorylation, deamination and cleavage of glycosidic bond like that of purine nucleotides to liberate the nitrogenous bases cytosine, uracil and thymine. These bases are then degraded to highly soluble products beta-alanine and beta-aminoisobutyrate. These are the amino acids which undergo transamination and other reaction to finally produce acetyl coenzyme A and succinyl coenzyme A. Dear students, I hope you could understand what are nucleotides, how they are synthesized and degraded. Thank you for watching. See you in next class with another topic. Don't forget to leave a like and please subscribe my channel.